There is a forest in the state of Oregon. It looks ordinary enough. Douglas firs, some undergrowth, birds moving through the canopy. But a few feet below the surface, something is alive that covers nearly 2,400 acres. It weighs an estimated 35,000 tons. It is, by some measures, the largest living organism on Earth, and it has no brain, no heart, no eyes. It does not move. It does not breathe. It has been growing quietly in the dark for somewhere between 2,000 and 8,000 years. It is a fungus, and the forest above it is not just sitting on top of it. That forest is, in a very real sense, dependent on it. Without it, many of those trees would die. Without fungi like it, without the entire hidden kingdom of fungi, most complex life on land would collapse within years. We live inside an ecological system that runs on fungi, and most of us have never once thought about it. That changes today. Fungi are one of the strangest success stories in the history of life on Earth. They are not plants. They are not animals. For most of human history, we didn't know what to call them, so we lumped them in with plants, and that was a mistake. Biologically speaking, fungi are closer to you and me than they are to a flower. There are an estimated 2.2 to 3.8 million species of fungi on Earth. We have formally described fewer than 150,000 of them. That means we have cataloged somewhere around 6% of the entire fungal kingdom. The remaining 94% is still out there, in the soil, in the ocean, on the skin of plants, inside insects, drifting through the atmosphere, completely unknown to science. What we do know is remarkable enough. Fungi help build the oxygen-rich atmosphere that made animal life possible. Fungi broke down the first forests when nothing else could. Fungi gave us bread, wine, beer, cheese, soy sauce, penicillin, and organ transplants. They are being studied as a potential treatment for depression, PTSD, addiction, and end-of-life anxiety, and they may become our most powerful tool for cleaning up industrial pollution and building sustainable materials. This is the story of the hidden kingdom, and it starts a very, very long time ago. Before we go anywhere, we need to settle one fundamental thing: what a fungus actually is. Most people, if asked, would describe a mushroom. Fair enough. But a mushroom is to a fungus what an apple is to an apple tree. It is the fruit, the reproductive structure, the part that pokes above the surface to spread spores into the air. The actual body of the organism, the fungus itself, is invisible. That body is called the mycelium. It is a web of microscopic threads called hyphae, branching and branching again, spreading through soil or wood or any organic material the fungus happens to be growing through. These threads can be impossibly fine, thinner than a human hair, but collectively they cover enormous distances. A single teaspoon of healthy forest soil contains miles of hyphae. Fungi don't have stomachs. They digest their food externally. They release enzymes into whatever material they're growing through, break it down chemically, and then absorb the resulting nutrients directly through their cell walls. It is a mode of eating that is deeply alien to anything we do, but extraordinarily effective. Here is the part that surprises most people. Fungi are more closely related to animals than to plants. The evolutionary split between the fungal lineage and the animal lineage happened roughly 650 million years ago. The split between fungi and plants happened significantly earlier than that. When you eat a mushroom, you're eating something more closely related to yourself than to the broccoli sitting next to it on the plate. This was not known until the late 20th century. Before molecular biology gave us the tools to compare DNA sequences across species, we had no way of knowing how things were truly related. Fungi looked like plants. They stayed in one place. They grew from the ground. So we assumed they were plants. The science of mycology was for a long time treated as a branch of botany. It took decades for the field to establish its own identity. The formal recognition of fungi as their own kingdom, separate from animals, plants, bacteria, and everything else, came in the 1960s and 1970s, when biologists began to understand just how different these organisms really were. We had been living alongside them for all of recorded history without understanding what they were. Fungi are old, older than the dinosaurs, older than the first land plants, older, arguably, than almost anything complex we can point to. The earliest confirmed fungal fossils date to around 800 million to 1 billion years ago. Molecular clock analyses, methods that use the rate of DNA mutation to estimate when lineages diverged, push the origin of fungi even further back, possibly to 1.3 billion years ago. For context, complex multicellular life wasn't even a settled experiment yet. About 450 million years ago, something enormous happened. Plants colonized land. This is often described as one of the defining events in the history of life on Earth, the moment when dry continents went from bare rock to green. And here is the part that rarely makes it into textbooks. Plants almost certainly did not do it alone. The earliest land plants had no roots. They had no way to extract nutrients from rock and soil efficiently. What they had, and what the fossil record strongly suggests, was a partnership with fungi. The fungal threads reached into the mineral substrate, extracted phosphorus and nitrogen and other nutrients, and delivered them to the plant. In exchange, the plant gave the fungus sugars produced through photosynthesis. This exchange, called mycorrhiza, is not a relic of ancient history. It is happening right now in the roots of roughly 90% of all plant species on Earth, including most of the trees in most of the world's forests. The partnership is so ancient and so fundamental that most plants can barely grow without it. Remove the fungal partners from the soil, and many plant ecosystems begin to fail. Life on land, as we know it, was a joint venture. Fungi were half the founding partnership. But the most dramatic chapter from ancient fungal history requires us to travel back to the Devonian period, roughly 400 million years ago, and meet something called prototaxites. If you walked across the land surface of Earth at that time, you would encounter a landscape almost beyond imagination. Low, mat-like plants, primitive mosses, early forests of small trees just beginning to establish themselves. And rising above all of it, reaching up to 8 meters tall, about 26 feet, were enormous pillar-like structures made not of wood, but of interlocking fungal threads. Prototaxites, the largest individual organisms on land at that point in history. Paleontologists debated for decades what they were. At various points they were classified as algae, as conifers, as lichens. It wasn't until careful analysis of their internal structure and chemical composition that the consensus settled on. They were fungi, enormous towering fungi, dominating a landscape that trees hadn't yet learned to fill. The world looked like that for millions of years. And then the trees caught up. You've probably heard the phrase wood wide web. It circulates through popular science articles with enough frequency that it risks becoming a cliché. But the underlying discovery is so astonishing that it deserves a careful telling. In the 1990s, a graduate student named Suzanne Simard was doing research in the forests of British Columbia, Canada. She was studying how trees competed for resources, how they fought for light and water and nutrients in the way that competition is traditionally understood in ecology. What she found instead was something entirely different. Trees were not simply competing, they were, in a meaningful sense, cooperating. Through the mycorrhizal networks beneath the soil, webs of fungal threads connecting root systems across the forest floor, trees were exchanging carbon, nitrogen, and water. Larger, older trees were supporting younger, smaller trees growing in their shade. When trees were stressed or damaged, other trees in the network showed responses. The network was not a metaphor. Carbon, labeled with radioactive tracers so it could be tracked, was literally moving from one tree to another through fungal channels. Simard began calling the largest, most connected old trees mother trees. They functioned as hubs in the network, exchanging more resources with more partners than smaller trees, and preferentially supporting their own offspring seedlings. The term has generated scientific debate. Some researchers argue it anthropomorphizes what are, at the end of the day, chemical gradients. But the exchange of resources is not in dispute. It is real and it is measurable. The fungi involved are not passive pipes. They are active partners. They regulate the flow. They take a cut in the form of carbon sugars for every transaction they facilitate. Some species of mycorrhizal fungi are remarkably selective about which plants they connect and under what terms. What we are looking at beneath the forest floor is not a quiet, inert substrate. It is a dynamic, living network of exchange, older than any economy, more complex than any market we have ever built. The forest is not a collection of individual trees. The forest is a community. This chapter has an uncomfortable premise, so let's just say it plainly. Without fungi, the world would be buried in the dead. Every organism dies, every tree falls, every leaf drops, every animal's body hits the ground. And all of that organic material, the cellulose in wood, the lignin in bark, the proteins in flesh, has to go somewhere. It has to be broken down and recycled back into forms that living things can use again. Animals can decompose some of this. Bacteria handle a great deal. But the toughest, most durable materials, the structural tissues of wood, the cell walls of plants, are largely beyond the reach of bacteria. They require something with more enzymatic firepower. Fungi have enzymes that can break down lignin, the compound that makes wood hard and durable. No animal can do this. Most bacteria can't either. The ability to digest lignin is, across most ecosystems, almost exclusively a fungal specialty. Here is a striking way to understand how important this is. About 300 million years ago, during the Carboniferous period, the forests of Earth were vast and lush and growing at enormous speed. Trees had evolved lignin as a structural material, but the organisms capable of breaking lignin down efficiently hadn't yet caught up. So when trees died, they didn't fully decompose. They fell, piled on top of each other, got buried, and over millions of years became coal. The coal we mine today, the coal that powered the Industrial Revolution, the coal still burned in power plants around the world. It exists because fungi hadn't yet evolved the right enzymes to break down lignified wood. When those enzymes evolved, the great burial of carbon slowed dramatically. We are literally burning the consequence of an ancient gap in the fungal toolkit. Today, the toolkit is complete. White rot fungi, brown rot fungi, and countless other decomposers work constantly, quietly, invisibly, breaking down the dead and returning their components to the living. They are the planet's recycling system. Without them, the cycling of carbon, nitrogen, and phosphorus through ecosystems would grind toward a halt. Nutrient flow would stall, soils would degrade, most plant life would eventually fail. The beauty of a forest depends on the invisible work happening in the rot on its floor. Not all fungi are benign partners. Some are killers. The story of the American chestnut is one of the great ecological tragedies in recorded history. At the beginning of the 20th century, the American chestnut was one of the most abundant and important trees in eastern North America. Roughly 4 billion trees, making up 25% of Appalachian hardwood forests. Indigenous and settler communities alike relied on the nuts for food. The timber was light, rot-resistant, and highly valued. The chestnut seemed, in the early 1900s, like a permanent feature of the American landscape. Then, in 1904, a shipment of Asian chestnut trees arrived at the New York Zoological Park carrying a fungal pathogen, Cryphonectria parasitica, the chestnut blight fungus. Asian chestnuts had evolved alongside it and were resistant. American chestnuts had never encountered it and had no defenses whatsoever. The blight spread at roughly 50 miles per year. By 1950, the American chestnut was functionally gone. 4 billion trees killed in fewer than 5 decades. Stumps still send up shoots, still trying, but the blight infects them before they can reach reproductive age. For practical purposes, the species exists as a ghost in its former range. Dutch elm disease did something similar to the elm trees of North America and Europe. White nose syndrome, caused by the fungus Pseudogymnoascus destructans, has killed tens of millions of bats in North America since it was first detected in 2006. Devastating bat populations that provide critical insect control across large agricultural regions. And then there is Ophiocordyceps. You may know it from the television series The Last of Us, which imagined a world where a fungal pathogen had evolved to infect human beings. The inspiration for that story was real, and the reality is strange enough to be unsettling on its own terms. Ophiocordyceps is a genus of fungi that parasitize insects, and in several species, the mechanism looks genuinely like mind control. A spore lands on an ant, penetrates its exoskeleton, and begins to grow through the ant's body. As it spreads, it somehow alters the ant's behavior. The ant abandons its colony. It climbs, always to a very specific height above the forest floor, determined by the fungus's preference for temperature and humidity. It clamps its mandibles onto a leaf vein, and it dies there, locked in place, while the fungus grows out of its head and releases spores onto the ants passing below. How does the fungus do this? It does not appear to invade the brain directly. Instead, it produces compounds that hijack the ant's muscular and nervous systems from outside the brain tissue. We are still learning how it works. What we know is that Ophiocordyceps species are extraordinarily specialized. Each species typically targets one specific host insect, and they have been doing this for at least 48 million years, based on fossil evidence of infected ants found preserved in amber. Life, for fungi, is not always a cooperative venture. Sometimes it is an arms race. You have eaten fungi your entire life. If you have ever eaten bread, you have consumed the work of yeast. If you have drunk wine or beer, fermentation by yeast converted sugars into alcohol. If you have eaten strong cheese, brie, gorgonzola, camembert, fungi develop the flavor and the texture. Soy sauce, miso, tempeh, all fungal fermentation. Vinegar, certain cured meats, sour cream. Many vitamins taken as supplements are now produced by fungal fermentation on an industrial scale. The relationship between humans and fermentation-producing fungi is so old and so fundamental that it predates agriculture. Evidence of intentionally fermented drinks, ancient beers and wines, goes back at least 9,000 years, and some researchers push the date considerably further. The specific species Saccharomyces cerevisiae, baker's yeast, brewer's yeast, has been living in close partnership with human civilization for millennia, shaped by human selection just as domestic animals were shaped by it. We co-evolved with it without ever knowing what it was. And then there is the truffle. Few foods on earth command prices like the truffle. The white truffle of the Piedmont region in northern Italy routinely sells for thousands of dollars per kilogram, sometimes significantly more. A single specimen of extraordinary quality can fetch prices at auction that rival luxury jewelry. The flavor, which develops from a complex cocktail of sulfur and other aromatic compounds, is considered by many chefs to be simply irreplaceable. Truffles are mycorrhizal fungi. They live in root partnerships with oaks and other trees, growing their fruiting bodies underground. That underground habit makes them extraordinarily difficult to find. Pigs were used for centuries. They can smell truffles through the soil with remarkable accuracy. The problem? Pigs also eat truffles when they find them. The modern solution? Trained dogs, which locate truffles with equal enthusiasm and less appetite. Attempts to cultivate truffles reliably at scale have met with limited success. The mycorrhizal relationship is delicate and depends on specific soil conditions, tree partnerships, and microbial communities we do not fully understand. Truffles remain largely a wild harvest, which is one reason they remain so expensive. The most prized ingredient in French haute cuisine is a fungus that we still do not know how to farm reliably. On September 3, 1928, Alexander Fleming returned from a vacation to find something unexpected in his laboratory. He had left Petri dishes of Staphylococcus bacteria, the kind responsible for dangerous infections, sitting out while he was away. One of the dishes had been contaminated by a mold spore that had drifted in through an open window, and around the contamination there was a halo, a clear ring where the bacteria simply were not growing. The mold was Penicillium notatum. The substance it was producing, penicillin, turned out to be one of the most important medicines in human history. Fleming's discovery sat in the literature for years, underdeveloped. It was only in the early 1940s, with the Second World War creating urgent demand for antibiotics that could treat infected wounds, that a team at Oxford, Howard Florey, and Ernst Chain developed methods for producing penicillin in useful quantities. By the end of the war, it was being manufactured by the ton. Infections that had killed soldiers throughout all previous wars were now survivable. The antibiotic era had begun, and its foundation was a fungus. This was not a coincidence. Penicillin was not some freak accident of chemistry. It was a weapon that Penicillium mold had evolved over millions of years to suppress competing bacteria in its environment. We borrowed a tool from an arms race that had been running long before humans arrived, and penicillin was just the beginning. Statins, the cholesterol-lowering drugs taken by hundreds of millions of people worldwide, were originally derived from a Penicillium species discovered in the 1970s. Cyclosporine, the drug that made organ transplants possible by suppressing immune rejection, came from a soil fungus discovered in Norway in the same decade. Various antifungal, antiviral, and anti-cancer compounds have followed. The soil beneath our feet, we are slowly learning, is an enormous untapped library of chemistry. Chemistry that fungi evolved over hundreds of millions of years for their own purposes, and that we are only beginning to read. How many more compounds are waiting in the 94% of fungal species we haven't yet described? Some fungi have played a different role in human history, one that is harder to measure and easier to misrepresent. Psilocybin is a compound produced by more than 200 species of mushrooms found on almost every continent. When consumed, it is converted in the body to psilocin, which activates serotonin receptors in the brain in ways that temporarily reshape perception, emotion, and cognition in profound ways. The experience is often described by those who have had it as one of the most meaningful events of their lives. How long have humans known about this? The answer is contested, but the evidence suggests a very long time. Mushroom motifs appear in prehistoric rock art in several regions. In Mesoamerica, the Mazatec people of Oaxaca have a ceremonial tradition involving psilocybin mushrooms, called Teonanacatl in Nahuatl, sometimes translated as flesh of the gods, that extends back centuries, possibly millennia. These were not recreational substances. They were tools of healing, of divination, of contact with the spirit world, used in specific ceremonial contexts under the guidance of trained healers. The healer who brought this tradition to the attention of the Western world was a Mazatec curandera named Maria Sabina. In 1955, an American banker and amateur mycologist named R. Gordon Wasson attended one of her ceremonies, the first outsider to do so with her cooperation. His account, published in Life magazine in 1957, brought global attention to what the Mazatecs had been doing quietly and privately for generations. The consequences for Maria Sabina and her community were difficult. Outsiders flooded in. The sanctity of the ceremony was disrupted. Sabina herself said later that once the knowledge left her people, the mushroom lost some of its power, not in any mystical sense, but in the sense that context, preparation, and intention matter enormously to the experience. By the early 1960s, the compound psilocybin had been isolated and synthesized. Researchers at institutions like Harvard were conducting early studies. Some of that work was sloppy and sensationalized. When the broader counterculture movement adopted psychedelics, the political response was severe. The Controlled Substances Act of 1970 placed psilocybin in Schedule I, the most restrictive category, reserved for substances deemed to have no medical value and high potential for abuse. Research essentially stopped. It stayed stopped for nearly 30 years. Then, slowly, something shifted. A handful of researchers at Johns Hopkins University, at Imperial College London, at NYU, began filing applications to study psilocybin in carefully controlled clinical settings. The early results were striking enough that the pace of research accelerated. The findings that have emerged since the early 2000s challenge assumptions that have been baked into policy for half a century. Psilocybin-assisted therapy has shown significant results in clinical trials for treatment-resistant depression, for alcohol and tobacco addiction, and for anxiety in people facing terminal cancer diagnoses. The FDA granted psilocybin a breakthrough therapy designation, a status reserved for treatments showing early evidence of substantial improvement over existing options in 2018 and again in 2019 for different conditions. The therapeutic mechanism, as current research can best describe it, appears to involve temporarily reducing the rigidity of habitual thought patterns, the mental grooves that keep people locked in depression, addiction, or grief, and allowing a kind of psychological reset. The experience itself, in the right conditions with proper preparation and integration, seems to create lasting changes in perspective. Whether this becomes mainstream medicine remains to be seen. The research is still in clinical trial phases. The regulatory, cultural, and logistical challenges are real and substantial. But the direction of the evidence is unmistakable. A compound that a culture in Oaxaca has used carefully for centuries, that the modern world criminalized and abandoned, may turn out to be one of the most important psychiatric treatments of the 21st century. The mushrooms were right. We just were not ready to listen. In a warehouse in upstate New York in the early 2000s, a company called Ecovative Design was growing furniture packaging, not manufacturing it, not stamping it from plastic molds, growing it. They took agricultural waste like corn stalks and seed husks, combined it with the mycelium of specific fungus species, and placed the mixture into molds. Within a few days, the mycelium had grown through the agricultural waste, binding it into a solid form. The result was a material that behaved like foam packaging, strong, lightweight, shock-absorbing, but was completely biodegradable. Put it in soil, and it decomposed in weeks. This was not a science fair project. The packaging went into commercial use. Companies like Dell and IKEA trialed it as an alternative to polystyrene foam. The technology has since expanded. Mycelium-based leather alternatives, mycelium-based acoustic panels, mycelium construction materials. In laboratories and startups around the world, researchers are exploring what else mycelium can do. There are fungi that can break down certain plastics. Pestalozziopsis microspora, isolated from the Amazon rainforest in 2011 by students at Yale University, can degrade polyurethane, a common plastic, even in the absence of oxygen. This is not yet a practical pollution solution, but it points toward possibilities. There are fungi being studied for bioremediation, the use of living organisms to clean up contaminated environments. Certain species can accumulate heavy metals from contaminated soil. Others can break down petroleum hydrocarbons. After oil spills, after industrial contamination, mycologists have begun experimenting with fungi to detoxify the damage. The same kingdom that built the foundations of life on land may be part of the toolkit for rebuilding what industrial civilization has damaged. Here is the thing we have been circling around for this entire journey. We tend to think of the natural world as something out there: forests, oceans, species, things we study, things we use, things we damage, things we occasionally try to protect, things that are separate from the systems we have built. Fungi do not fit comfortably in that frame. Fungi are not a part of the natural world that we discovered. They are the substrate on which almost everything we have built is resting. The bread on your table, fermented by fungi. The antibiotics that kept previous generations alive long enough to have children, from fungi. The forests that maintain the water cycles and carbon cycles that make agriculture possible. Mycorrhizal fungi made those forests. The soil itself, built over millennia by fungal decomposition. The psychiatric medicines that may reshape mental health treatment in the coming decades, partly from fungi. We didn't choose to depend on fungi. We evolved into a world that fungi had been running for hundreds of millions of years before our species arrived. The partnership between plants and mycorrhizal fungi is 450 million years old. Fungi were building towers on the landscape before trees had figured out how to. We arrived late in an ecosystem that fungi had been fundamental to from the beginning. And for most of human history, we didn't even know they were a separate kingdom. We called them plants. We ignored them in our models of ecosystems. We criminalized the ones that generated the most interesting neurological effects. We burned the compounds they produced in the form of coal without knowing what we were burning or what had created it. The hidden kingdom was not hidden because it was small or inconsequential. It was hidden because we weren't looking. We are starting to look now. There is a concept in mycology called the dark matter of biology, a phrase borrowed from physics, where dark matter refers to all the mass in the universe that we can detect only by its effects, because we cannot yet observe it directly. Mycologists use the phrase for the vast majority of fungal species we know exist but have not yet described. Millions of species, each with its own chemistry, its own relationships, its own ecological role, its own potential uses, we cannot yet imagine. We are right now in the early chapters of understanding what fungi can do. The pace of mycological research has accelerated dramatically, driven by new genomic tools that allow scientists to identify fungal species from soil samples without ever seeing them, by the psychedelic research renaissance, by the search for new antibiotics as existing ones lose effectiveness against evolving bacteria, and by the growing urgency around environmental remediation. What comes next is genuinely unknown, possibly new medicines we cannot currently conceptualize, possibly materials that replace plastics in ways we haven't designed yet, possibly new understanding of how forests work that changes how we manage land, possibly new treatments for diseases of the mind that we have struggled with for generations. The oldest kingdom is, in a sense, the newest frontier. The forest floor beneath your feet is not static. It is not dead. It is alive with exchanges we are only beginning to read. Transactions in carbon and nitrogen and water, conversations in chemistry between organisms that have been in conversation for hundreds of millions of years. We are newcomers here. Fungi were building this world before we arrived. They will be here long after we are gone. The wisest thing we might do is pay attention to what they have already figured out. If this journey into the hidden kingdom left you hungry to know more, consider subscribing. There is so much more to explore.